Hello to everyone. Thank you for coming, and we have English participants today, so I think we should perform it in English. Today we have the talk of Alexei Galatenko and his colleagues, which is entitled as Mathematical Models of Within Host COVID-19 Dynamics. And Alexei, please, you can start.
Thank you very much. First of all, I'd like to thank the organizers of the seminar for the outstanding possibility to present our results. And I want to especially thank Professor Wolpert for his outstanding talk. Well, it was a pleasure to understand that some ideas found in the process of our research were also found by other people, well, which are great specialists in the fields. It was an argument that our road is a correct one. All right. Professor Wolpert considered COVID-19 at the scope of population. The objects under consideration were individuals, healthy, susceptible to the virus, infected or recovered after the infection. We will discuss the process of infection within host. We're dealing with cells, infected or uninfected, viral particles, and so on. Why is this problem interesting, especially from the practical point of view? Well, there is a number of simple models which consists of two or three differential equations that describe the disease dynamics and give the researchers an opportunity to find answers to some interesting questions. For instance, in the paper by Iwanami and co-authors, the authors performed the comparative analysis of treatment efficiency. They used the mathematical model to find out when to give treatment and what treatment is optimal for COVID-19. And another paper of the same group, the first author is different, but actually these are the papers by one large research group. In the second paper, the author tried to justify the isolation period, to use mathematical model to find out the time after which the number of viral particles in the host drops below the threshold, so the person can be considered recovered. Another interesting paper by the same group of researchers considered the problem of locating the disease source. Given the symptom onset time and possibly some other measurements, for instance, viral load at some point of time, they try to find out when the virus entered the host. If they successfully solve this problem, then they can locate the source, the infection source, and thus, well, isolate it and reduce the number of infected people. All right. The key problem in this research is to fit the model parameters to real data. In practice, the researchers just used known tools like dbinfer, which is an R package, or Monolix, which is a standalone tool, freely available for research purposes. Well, they just took the model, took the data available, run the parameter fitting software, and use the solution, use the answer returned by the software. So that's it. And after parameters are determined, one can investigate the model, for instance, by numerical modeling. We have differential equations, and we can solve them using one of well-known numerical methods. As a matter of fact, analytical results on the solutions of systems of differential equations that model infection dynamics to the best of our knowledge are sparse. And on the other hand, since our model is simple, since we have just 2 or 3 parameters, many properties which may be essential from biological point of view are lost or just blurred. Examples of such blurred properties will be presented a little bit later. All right, so there are simple models, but there are also very sophisticated models. They consist of up to dozens of equations. As a result, reliable identification of parameters using existing data seems to be a hard problem. And obtaining analytical solutions seems to be a very hard problem. As an example, there is just a teeny part of a model consisting of 76 equations. You can see that they are nonlinear. They have many variables. Well, they look fearsome. The authors of the paper cited here also took a standard software package, somehow determined the parameters, made conclusions, but the question is, how reliable is this parameter identification? I do not have an answer to this question. All right. In the framework of our talk, we plan to discuss 3 points. First of all, we'll talk a little bit about analytical results on simple models. Then we will discuss available data which can be used to tune our models. And finally, we'll present a moderately sophisticated model that, from our point of view, adequately explains data obtained for cell line experiments. All right, let's proceed. The canonical system in 3 variables that describes within-host dynamics is the following. Let U be the number of uninfected cells, I be the number of infected cells, and V be the number of free variants. We assume that the healthy cells get infected proportionally to the product of the number of cells and the number of free variants. Well, uninfected cells become infected cells, and infected cells may die with some intensity delta sub I. Viral particles are produced by infected cells with constant intensity P, and viral particles also die with intensity delta sub V. Here is a sort of a canonical model, which is considered in many papers devoted to virus dynamics. For instance, the one cited here at the bottom of the slide. Let's see what is blurred here. First of all, the death of infected cells and viral particles implicitly includes immune response. T cells kill infected cells, and antibodies kill viral particles. So there is natural death and there is immune response, and everything is messed up in just one parameter. The second problem was pointed out by Professor Volpert. The infected cells do not start viral production instantly. It takes some time for the production to start. So the intensity here should depend on the number of infected cells, not at the point T, but at the point T minus delta T of some previous point in time. There is another issue with the canonical model. Right, let's move on. In some experiments on cell lines, it was noticed that infected cells do not die. there is a link to the paper describing the experiment where the number of alive cells was measured, and this number was basically constant. Besides that, the death rate of variance is also negligible. The experiment is short, 48 hours, 72 hours, well, 96 hours, and within such a short period of time, one can consider death rate of variance to be equal to 0. It's negligible. So there is a simplified model in 3 variables. Here is the original model, and we set death rates equal to 0. The model is essentially simplified, so we will be able to obtain theoretical results on this model. Another simplification is the system in 2 variables. It was originally introduced in the paper by Ivanami, cited a little bit earlier. This paper analyzed various treatment strategies. The authors of the paper were unable to identify the parameters of the system in 3 equations, at least what they wrote in the response to the reviewer. So they decided to perform additional model simplification. They made a quasi-steady state assumption on the number of variance, and this assumption allowed them to obtain the system in 2 variables. Here, U is not the number of uninfected cells. Here, it is the fraction of uninfected cells. They performed normalization of the cell quantity. And V, again, is the number of viral particles. So the fraction of healthy cells gets reduced proportionally to this product, and viral particles are produced with an intensity which is proportional to the product of the number of healthy cells and viral particles, and die at some death rate delta. All practical results from the second slide are based on this system. Moreover, at the conference on mathematical methods in biology, there were talks based on this model in 2 variables and 2 equations. So this model is, well, sort of widely used. Now let's proceed to analytical solutions of this system in 2 variables and 2 equations. In order to formulate our results, we will need Lambert's W function. It is defined in the following way. Assume that we have a function f of x equal to x times exponent of x. The left figure is the graph of this function. Lambert's function is the inverse of f. That is, w of y times exponent of w of y is identically equal to y. Obviously, this function is multivalued on the interval from -1 divided by e0. It has 2 branches denoted by w sub -1 and w sub 0. The selection of the appropriate branch is determined by the context. For instance, in one case, the function is growing. The second case, it is decreasing. From the qualitative characterizations, we can select which branch is appropriate in which situation. All right. As I've already said, we assume that the point t0, t equal to 0, at the point of symptom onset, the number of uninfected cells is equal to 1. If not, we can just change the scale. And the number of free virions is equal to some constant v sub 0. It can be easily proved that the following equalities hold. These are just technical equalities that allow one to come to, well, reasonable and practically meaningful conclusions. The first equality expresses the number of viruses in terms of the number of uninfected cells. The second equality is a sort of the inverse of the first one. It expresses the number of uninfected cells in terms of the number of viruses. Finally, the third equality establishes the connection between the number of viruses and time. It expresses the independent variable t in terms of the dependent variable v. Well, it's not the expression of our dream. The inverse expression would be the expression of our dream, but still it allows one to come to interesting conclusions. Right. A very important property of the solution of the system of equation is the maximal viral load, Vmax, the maximal value of the number of viruses. Assume that this peak viral load is achieved at the time t sub max. This is the time when this viral load is achieved. These two parameters play an important role in the research by Ivanami, for instance. It determines the optimal time of treatment. Assume that H is some threshold which defines the healthy state. If the number of viruses is below this threshold, V sub H, then we say that the person is healthy. The number of viruses is negligible. And T sub H is the recovery time, the time when the number of viruses drops below this threshold. The next point is just some technical notation. And the assertion, the meaningful assertion, in contrast to the previous assertion, which is more like a technical one, allows one to express the number, the peak viral load, the time at which this peak viral load is achieved, and the time of recovery in terms of the initial parameters, in terms of the initial viral load and model parameters like beta, delta sub v, and so on. This is the explicit formula for these values. We do not have to solve the system of equations. We have an explicit expression. All right. And the assertions presented above also allow us to give the more of a qualitative characterization of the solutions. If delta is less than gamma, which means that virus production rate is greater than virus death rate, then the number of viruses first grows and then monotonously decreases. Otherwise, if death rate is greater than production rate, then it just monotonously decreases. And as t approaches infinity, the number of viruses tends to 0, which means that in the framework of the model, we will definitely recover. And the number, the fraction of uninfected cells approaches the constant, well, written out here. Here is the illustration of this behavior. The number of uninfected cells, well, first drops and then, well, goes to a asymptotic value. And if production rate is greater than death rate, we first get worse and then get better. And if production rate is less than virus death rate, if our immune system is very strong, then we just get better. We just clean the viral load and that's it. Here are some more analytical results concerning the simplified system in 3 variables. We're just in the process of solving this problem, so this result is far from the ultimate one. However, well, we decided to share it with you. C sub 3 here is some constant determined by initial conditions. Similarly to the previous case, the number of cells equals to 1. I plus U is equal to 1. And we can write out the expression for the number of viruses in terms of T. The number of infected cells in terms of V. And the number of uninfected cells also in terms of V. Well, the behavior of the system is much more complex than the behavior of the system in 2 variables. So we don't have such neat characterization and such neat pictures as in the previous case. If we consider the dynamics for longer periods of time, then the models considered above are no longer adequate, because we need to take into account healthy cell regeneration. There are several models of cell regeneration in the literature. For instance, the exponential growth or logistic growth or the mix of both. Anyway, if we add such a summand to the first equation, we'll get an even more sophisticated solution, the class of more sophisticated solutions. For instance, there emerge periodic solutions, which may correspond to long disease, long, like several waves of disease. And one of the problems that we want to solve is finding adequate parameters under which the disease will go in several waves. The link to the paper is link here describes various ways of modeling cell regeneration. In some conditions, well, the authors advise to use one model, in some conditions another model. It depends on the disease type and on the well, model under consideration. For instance, the situation for cell lines and the situation for in vivo experiment are different. All right, let's turn to the second subject of our talk, the data which can be used to fine-tune the model. If we talk about in vivo models, then in the vast majority of research, we have just one source of data, which is RT-qPCR analysis data of patient swabs. That's it. But if we also consider in vitro experiments, then we get essentially more data available. We definitely know the initial number of cells and the initial number of variants. We definitely know the time of infection, which is the time at which our experiment started. We can measure, well, the researchers can measure variant quantity, normally using TCID sub 50 measurement techniques. We can also measure the concentration of intracellular virus RNA, for instance, by RT-qPCR analysis, which compares the viral load with the housekeeping gene, GAF-DH. We can also find the number of alive cells at the end of the experiment. we can additionally measure the number of infected cells at some points of time. Well, this is impressive. A whole lot of various types of data which can be taken into account. Here is an example of data used in vivo analysis. Well, it was used in the paper which was already cited several times. First of all, these are just pictures. And as far as I remember, the authors used the utility data thief to convert these pictures, these figures, into numerical values. Second, obviously, this data is noisy. For instance, take a look at Singapore 9 picture. There are several measurements pointed by red dots, which were definitely a failure. It is highly unlikely that viral concentration dropped to 0 and then returned to the high value once again. Well, nothing else was used by the authors of this paper to perform analysis. All they could do is to fit their model to such noisy data. And if you take a look at these pictures, you will see that fitting is far from perfect, especially take into consideration the fact that this is a logarithmic scale. So the difference between some points is the order of magnitude. It's not just 1, it's 10 times. So the quality of fitting to such noisy data is also a sort of the point of doubt. And here is the example of data collected in vitro experiment by Shuai and co-authors. The authors considered 2 virus strains, the original Wuhan strain, wild type, and Delta strain. And they used KCO2 cell lines in their experiment. And here are the measurement results. Here are the left part of the figure is the viral concentration, and the right part is the viral intracellular viral RNA concentration. The second source of data is the viability assay, and from this picture we can make a conclusion that all cells are viable. Cells do not die throughout the experiment. And the authors additionally measured the fraction of infected cells at t equal to 24 hours post-infection. And this fraction was very small, just 6%. And in private communication with the authors of these papers, we found out that this number, I mean 6%, holds for, well, the published 6% value holds just for the wild type. But this is a typical number for other strains as well. And the researchers also confirmed that viral death is negligible. We can say that viruses do not die throughout the experiments. Well, they do die, but the death rate is very small. Well,
excuse me,
yeah.
may I ask a question? These experiments about virus death, they're in vitro. Am I
Yeah,
understand
exactly.
right?
Yeah, KCO2 cell lines. And the duration of experiment is just 48 hours. We just do not say that viruses do not die at all. They do not die fast. This is what we say. Actually, if you look at this picture, viral load becomes more or less reasonable after, well, 24 hours after infection. So what we say is that viruses do not die within 24 hours.
Mhm.
This
And
is a much less
it means
strongest. I'm
it
sorry.
means some kind of natural death of virus and not the death because of immune response. Am I right?
Yeah. There is no immune response in the cell line. There are no immune cells, and no antibodies.
And
The data is much less noisy.
I have one more small questions about the in vitro experiments. Maybe you know if there are any techniques, are there physiological experiments possible in vitro with respect to immune cells?
Unfortunately, I do not know the answer to your question. If we talk to Dr. Chu once again, I'll ask him this question.
Thank you. If you know the answer, please let me know as well.
Sure. He's a
Thank you.
magician of experiments. Well, very impressive, very interesting experiments, very interesting results, very interesting data. Thank you, Dr. Chu. I know that the word is magic. It will reach you. All right.
Yeah, thank
Let's
you.
move
Please.
forward. So if we switch from an in vivo model to an in vitro model, we'll have a number of benefits. The first one, we have a substantially greater volume of data. The cells in the cell line are much more homogeneous in comparison with a human being, as there is no adaptive immune response. There are no innate immunity cells in the cell line. There is the data is much less noisy. The experiment is reproducible. We can take yet another copy of a cell line, another portion of viruses, once again infect the cell line and reproduce the experiment. In case of humans, well, reproducing is quite challenging. However, to the best of our knowledge, mathematical models of in vitro dynamics were almost unconsidered in the literature. We have found just one paper cited here, and even this paper was solely based on viral load data. It did not take into consideration other types of data available. Well, before formalizing our model, let's take a look at the data and make some conclusions. First, as I have already pointed out, like 20 times, all cells stay viable throughout their experiments. Second observation, take a look at the dynamics of growth of viruses and intercellular RNA. You can see that first, this growth is very fast, up to 24 hours, and then it essentially decreases. Growth rate decreases over time. And the fraction of infected cells at t equal to 24 hours is relatively small, just 6%. So decreased growth rate cannot be due to the exhaustion of uninfected cells. There is a whole bunch of uninfected cells. What is the reason of the decreased growth rate? Simple models are unable to reproduce such a behavior. We need to think of a more sophisticated model that adequately explains this data. Well, our hypothesis made together with the biological part of our team are the following. First of all, infected cells and virions do not die. Well, the second one is that the decreased growth rate of the number of viruses is due to the exhaustion of some intracellular resources, for instance, lipids. It takes some intracellular resource to produce a virus, to produce a virus coat so it can leave the cell. And we hypothesized that well, the number of such resources is finite, and after some point, this resource gets exhausted. And the growth rate of intracellular viral RNA decreases due to decreased infection rate caused by immune response. For instance, it can be initiated by cytokine emission. Well, here is the description of our model in terms of a picture. When virions enter the cell culture, they infect some healthy cells. After a cell gets infected, it starts production of viruses with some rate P of t. You see, here P depends on time. Viral production rate is not a constant. And virions can enter either infected cells with some decreased intensity. This is a known phenomenon. If a cell is infected, then the intensity of infection drops. It can also enter a healthy cell with a higher rate, or an immune cell with still lower rate. To simplify the model, we assumed that infection intensity is equal for immune cells and infected cells. This was done just to reduce the number of parameters. At the qualitative level, it worked perfectly fine. All right. And in the process of producing viruses, the infected cell exhausts its resources and either becomes exhausted or even dies. And another function is that an infected cells produces cytokines with some constant rate and some delay. And these cytokines enter healthy cells and make them resistant. This is our model from the qualitative point of view. Let us switch to the...
Excuse me.
Yeah.
Could we return back to the and one more back? I didn't understand well about the virion growth rate decreases due to exhaustion of some intracellular resources. Do you mean that there is exhaustion of resources of infected cells?
Yeah.
Because there is no exhaustion of
Exactly.
uninfected cells.
Exactly. The infected cell gets exhausted and is no longer able to produce viruses. So the production rate drops.
So the infected cells in your models does not produce viruses all the time.
Yeah,
and
this function P of t after some time becomes equal to 0. So the production stops.
If there is some differences between the exhaustion of intracellular resources, from the modeling point of view, if there are differences between infected cells which are not producing virus anymore and dead infected cells, can we just increase the death coefficient?
Well, in our model, death rate is equal to 0. So the difference is that there are no dead cells in our models, but there do exist exhausted cells.
Okay, I understand. So you have some term negative, which you interpret as exhaustion of infected cells, but not death.
No, actually, our solution was very close to the one proposed by Professor Volpert in his talk. Just take a look at the 1, 2, 3, 4, the fifth equation here. You see, we take the number of cells infected at time t minus x, multiply it by p of x, which is the virus generation intensity, and then we integrate this value by x. So we explicitly use this kernel.
Okay, thank you.
This kernel
Please,
specifying virus generation intensity.
I interrupt you on the description of your questions, please.
Thank you very much for the
Continue.
questions. It is an honor for me to understand that, well, people do listen to the stuff I'm talking about. Thank you very much. So here is the system in 6 variables and 6 equations. U0 is the number of regular healthy cells. And a healthy cell can either get infected with intensity beta. This is similar to the term from the simple models, or it can be made resistant. Here, just the viruses are replaced with cytokines. And this term is also very similar to the ones considered in the simple models. And what happens to resistant cells? They emerge from regular cells, and they can be infected with some smaller intensity. K is the parameter determining the reduction in infection rate caused by such immunity. Here, the third equation corresponds to cytokine production. They are produced with constant rate with a certain delay. And they are used to make regular cells immune. The fourth equation is corresponds to infected cells. They are produced either from regular healthy cells or from resistant healthy cells. Probably the fifth equation is the most interesting one, and it is in sync with the proposal made by Professor Volpert in his talk. Let us integrate the values using some kernel. In this case, we can take into consideration the delay, which is caused by the fact that virus production does not start instantly, and resource exhaustion. After some time, P will decrease and finally become equal to 0, so production will stop. This is the source of virus generation. The expression in parentheses is just I prime. You see, the expression in these parentheses is just I prime. And viral particles, they do not die, but they are used to enter either infected cells or uninfected cells or immune cells. The sixth equation is for intracellular RNA. On the one hand, it is similar to the fifth one. Viral RNA is produced in a similar way using another kernel, sigma of x. On the other hand, this variable r of t is not used anywhere, but it is measured. It is measured in the experiment. So this equation was added solely to match data, to fit our model parameters to data. Otherwise, we would remove it.
May I ask one more question?
Sure.
This is a viral RNA intracellular or
Right, intracellular, the stuff that is stored inside cells.
and how it can be measured in experiments? How do they measure it?
They measure the number of the concentration of one of viral genes as G, and compare it to the concentration of GAP-DH gene, which is a
Oh.
housekeeping gene. It is a constant. Its expression is constant in cells, and we divide one value by another and obtain the concentration of viral RNA inside cells.
So in the experiments, they measure viral RNA inside cells, not outside
Right.
cells.
They measure viral particles using TCID 50 assay, and they measure intracellular viral RNA using quantitative PCR, I'm sorry.
Thank you.
All right. Now let's switch to model fitting. We performed parameter fitting by component-wise gradient descent. And here are the results. This dropped value of viral concentration is caused by washing. After, I think, 4 hours or 2 hours, I don't remember the exact values, the media was washed, so all viral particles were removed. And it stayed equal to 0 until new viral particles started to be emitted by the cells, by infected cells. The left-hand side corresponds to the Wuhan strain, and the right-hand side corresponds to the Delta strain. As you can see, our curves produced by the solutions of the system of equations with the parameters found using this gradient procedure, well, it adequately reflects the behavior of the model. You see that in the second case, the intensity is smaller. The number of infected cells is smaller, and the number of viral particles is also smaller. If we take a look at the parameters, I decided not to write them out. Maybe it was a bad idea. It is adequately reflected by the model. In the second case, in the case of Delta, infection intensity is less than for the Wuhan strain, and cytokine production rate is greater than for the Wuhan strain. So the cells are more protected and less in two senses. These results stay in sync with biological argument. It is known that intestinal complications are much less common for Delta strain, and KECO2 cells are the ones taken from intestinal. And finally, I still have 12 minutes, and I want to take a little survey of a discrete model of the same process. We also developed a discrete model, which also contained a number of continuous components. It was done with two goals. The first one is to perform model validation, to compare the results of a differential model and a discrete model. And the second goal is to create a model which is very intuitive, which is, a sort of obviously correct from biological point of view. So the main constituents of the discrete model are state components. There is a discrete state component, the number of regular cells, the number of resistant cells, the number of active infected cells and the number of exhausted infected cells. And we also have a continuous component of the state. First of all, the global part. This is the number of free virions and the number of cytokines. Continuous means that these are not integers. These values can be real. This was done to improve model efficiency. We do not model V and site at particle level. We just store the integral value. And we also have a local part of the continuous part of the state. It corresponds to infected cells. And each infected cells is assigned with 3 parameters. The time of infection, the number of viral RNA, and the number of intracellular resources. L stands for lipids. We hypothesize that these resources are in essence lipids. And the model works in discrete time. At some time moments, T1, T2, and so on, there occur events that change either local or global state components. At the time moment t equal to 0, which is at the initial time moments, all cells are regular. The number of virions is equal to some constant v sub 0. There are no cytokines and no viral RNA, and the number of resources in each cell is equal to some predefined constant L sub 0. Well, the cell is full of lipids. There are 5 regular types of events and 1 outstanding event. The outstanding event, the event of type 0, is washing. At the time moment T sub clean, the number of virions and the number of cytokines is set equal to 0. Everything is washed away from the media. And now let's switch to the regular events. The events of type 1 is making a regular cell resistant. A cytokine enters a regular cell and makes it resistant. Event type number 2 is making a regular cell infected. A virion enters a regular cell and makes it infected. Type 3 is infecting a resistant cell, and type 4 is sort of wasting a virus. It just enters a cell that is already infected. Well, just get wasted. And the 5th state is updating the system state. Like, we compute the number of virus particle emitted by cells, compute the number of cytokines emitted by cells, and so on. All events except of the 5th one can be implemented with a constant complexity. So this model can be implemented very efficiently. And the events of type 5, updating the system state, can be implemented with complexity, which is linear in the number of cells. We're talking about cell lines. A typical number of cells is 30,000. It's not that great. It is something that can be efficiently computed. All right. Now we need to select the time and the type of the next event. Event of the type 0 is fixed. It occurs at time t clean. And for the types 1 through 5, we compute the intensities. In the first case, making the cell immune, you see the term here corresponds to the term in the system of equations. The cell is made immune, is made resistant with intensity which is proportional to the product of the number of regular cells and the number of cytokines. This is the intensity of infection of a regular cell. The same thing as in the equations. And it also holds for infection of a resistant cell and for entering into an infected cell. Just recall that IA plus IE is just the number of infected cells. And finally, recomputation of the state is performed with fixed intensity. Now, consider T, let it be the moment of the current event. We want to select the time of the next event. The offset is set equal to delta sub T, which is computed in the following way. This is just the mathematical expectation of an exponentially distributed random variable that corresponds to total intensity of 5 flows. And the type of the event is selected randomly with probability proportional to the intensities. This model, in essence, is the variation of a well-known Gill-Spy model that allows to perform such event-based modeling. Here are the results of fitting the model to real data. These results are in sync with the results obtained for the differential model, with one exception. Here we have the values for the number of active and exhausted cells. We spent more time to tune this model, but we managed to get more data. We managed to explicitly find the number of active cells here. 2 minutes left. Okay. And future research. We want to continue analytical investigation of simple models. We're not great specialists in differential equations, but we learn quickly. We want to find realistic parameters that describe the disease dynamics consisting of several ways of infection. And we want to continue our research of in vitro models. There are data for new cell lines, for new virus strains, and we can fit our model to these data as well, and to perform a sort of a comparative analysis. Here are 2 slides with bibliography, 3 papers on application of the model into equations. The paper describing a huge multi-compartment model of 70 plus equations. The paper on the canonical system in 3 variables. The paper on the term that corresponds to cell growth for the regeneration of healthy cells. Here are 3 papers describing the data obtained from in vitro experiments. The paper on in vitro analysis using just viral load. And finally, our paper on the, well, moderately sophisticated model discussed in the 3rd part of our talk. Thank you very much.
Thank you very much for the presentation, Alexey. And if there are any questions, please.
Well, yes, Anastasia, if you allow me to start, because I'm in the process of studying intra-host dynamics of viral infection. So because of that, I cannot stay for a long time. I'd like to make some comments, and then I will leave you to continue the discussion. So thank you again, Alexei, for this very interesting presentation. And in fact, there are many questions which we worked on or thought about. So I think that, well, myself and other colleagues present here are interested by that. During the last several years, we have done a cycle of works on this infection dynamics, intra-host infection dynamics. We have more concentrated on the spatial effects. So we, well, you started with this, for example, ordinary differential equations. We were more interested in partial differential equations, where we take into account the spatial distribution of all these infected cells, uninfected cells, virus particles, and so on. But many of the questions which you mentioned, for example, like death rates or cell regenerations or intracellular proteins and cell exhaustion or interferon production and this innate immunity, many of these questions, in fact, we were thinking about, and in fact, they are very interesting and very important. Another, of course, important question you mentioned and discussed, it's really very, very interesting, and you have very, well, some new results for us and some new approaches related to comparison with the data. We also try to do some of such comparison. So, so I think that, of course, we cannot discuss all these questions in detail now. It would be too long. So what I would suggest, we can send you some of our recent publications on related topics. If there are some questions which seem to you interesting for further discussion, we can return to them and we can discuss them later in more detail. So
thank you very much.
This is what I think probably a possible way to continue this discussion, which seems to me very interesting and very promising. Well, unfortunately, I have to switch off now. I suppose that if Anastasia or other participants have questions or comments, then you can continue. Thank you very much again, and I hope we'll continue our collaboration and discussion.
Thank you very much.
Yeah, bye. Thanks.
Bye.
Thank you very much. Bye. Okay, if there are any other questions with the speaker, I have one small question about the, you mentioned that you have the biological part of your team. So do you mean that you in your team perform the physiological experiments?
Unfortunately, our specialist in biology just perform, well, I don't know how to say it in Latin, in paper research. They analyzed a whole bunch of literature and consulted us on what is biologically meaningful and what is biologically meaningless. The experiments, all the experiments discussed today were performed by Chinese group located in Hong Kong. And we had several discussions with Dr. Chu, who is perhaps the leader of this group. Well, fantastic experience. Said, well, tell us, what experiments are need to be executed? We'll do it promptly. That was fantastic.
So they perform the experiments as a result of your
unfortunately not. Unfortunately not. We're not insolent enough to say, okay, we need 10 experiments. Write it down. The first one, and so on. We are very polite people.
Yes. And thank you. As Professor Wolfert said, we have some works in this area, and it is very interesting. I think he will send you the papers, and we can discuss in more details, more I think more particular questions.
Thank you very much. Looking forward to it.
If there are any other questions to the speaker. If no, I think we can stop for it now and waiting for our further collaboration. So
Thank you very much.
thank you very much for
See you.
your presentation.
See you.
Bye.
Bye-bye.
Thank you. Bye.
Thank you. Goodbye.